Welcome to this explainer. Today, we're going to crack the code on how to elevate your Cambridge biology answers from average to absolute top tier. I am so thrilled to guide you through this because we are doing something a little different today. We aren't just reviewing biology, we're actually reviewing the exact framework examiners use to grade you.
So let's dive into this mystery right away. What exactly separates a B grade from an A star? Well, the difference isn't just about memorizing facts. If you've been memorizing textbooks and still falling short, it's really about how you structure your scientific reasoning. It's the difference between simply knowing the science and knowing how to communicate it perfectly to the examiner.
Here's our roadmap for today's explainer. We'll be covering the high-mark exam strategy, jumping into case studies on cell structures, core genetics, and biological processes, then highlighting top examiner red flags and wrapping up with some final exam takeaways.
Okay, Section 1, the high-mark exam strategy. We're unlocking the method. Let's establish a bulletproof framework you can apply to literally any syllabus topic.
This brilliant four-step framework is your anchor, and I really want you to burn this into your memory. Always start with concept identification. Then, extract specific evidence and data. Next, link them with scientific reasoning, meaning you explain the why. And finalize your answer with precise examiner wording. We call this the logic loop, and it's exactly how you secure those crucial method marks.
Moving right along to Section 2, case study on cell structures. Let's see how this framework builds when we look at microscopic cell architecture.
Before we even analyze a question, we absolutely must define our biological principles precisely. Generic definitions just won't cut it anymore. Notice how we don't just say the rough endoplasmic reticulum makes proteins, we highlight its structural and functional requirements. It's a membrane-bound organelle covered in ribosomes, acting as the primary site for high-volume protein synthesis specifically required for secretion.
When breaking down the A-star blueprint for mastering the RER, you'll find that top-band answers explicitly connect that massive surface area directly to high-volume production. A top candidate doesn't just say it has ribosomes. No, they specify 80S ribosomes and use comparative connectives like whereas to contrast RER-rich cells with SER-rich cells. That specific causal link is exactly what the examiner is hunting for.
Have you ever made the mistake of giving a generic fact instead of a specific context-driven response? Writing something like, the ER makes proteins, is incredibly flawed and gets you 0 method marks, because it completely lacks biological depth. On the flip side, the top-band answer stating the RER facilitates high-volume protein production required for secretion is precise, contextual, and hits all the required syllabus terminology.
The Golgi body is another brilliant example of where deep analytical reasoning wins out over superficial knowledge. This is a high-yield topic worth a massive 15% of paper 2. Marks are awarded for showing logical, data-driven modifications. A flawed answer just states, the Golgi processes proteins, but the master student writes, the Golgi modifies proteins by adding carbohydrate chains to form glycoproteins. And they actually reference the provided evidence, like radioactive labeling. Do you see the difference there? They linked the biological fact directly to the data.
Let's shift gears to Section 3, case study on core genetics. Applying our strategy to the molecular level is absolutely critical because the stakes are just as high here.
Mastering genetics requires incredibly precise definitions. Why? Because generic textbook answers simply won't earn you the method marks. You have to be meticulous. Every single word matters. The process involves the separation of strands, where each original strand acts as a template for the synthesis of a new complementary strand.
Applying the high-mark framework to semi-conservative DNA replication reveals something super interesting. Students constantly confuse their key enzymes. It's a huge terminology slip-up. Top-band answers explicitly state that DNA polymerase catalyzes the formation of phosphodiester bonds, while DNA ligase joins Okazaki fragments. Precision is definitely your best friend here.
So, the crucial point to take away is that writing DNA copies itself is a massive leak of easy marks. Honestly, it's a middle school answer. The top band answer explains the actual biological mechanism step by step, involving the separation of strands and synthesis of new complementary strands. Never assume the examiner knows what you mean. You have to explicitly prove it on the page.
Similarly, when dealing with the random fusion of gametes and genetic variation, examiners are basically begging you to extract specific phenotypic data from the stimulus table to ground your reasoning. A major red flag here is vague contextualization. If the exam gives you a specific organism or a specific genetic data table for an F1 generation, your answer must incorporate those exact figures and contexts. Don't just recite a generic definition of genetic variation. Moving on to Section 4, Case Study on Biological Processes. Let's explore dynamic biological processes and how we must handle data and statistical tools in our answers.
Here is a quick golden rule for processes. Always identify and establish your conceptual anchor before you start crunching the numbers provided in the exam. For instance, if the question is about enzymes, anchor your answer in the enzyme substrate complex before you try to explain why the rate dropped at a certain pH.
When investigating enzyme-catalyzed reactions, top candidates never just say, "It got warmer." That is a classic generic trap. Instead, they state specific temperatures, like increasing temperature to 40 degrees Celsius, and they explain how the increased kinetic energy of specific enzymes leads to more frequent successful collisions. It's always specific value, correct terminology, precise outcome.
Let's pause on statistical tests for a second, because knowing which tool to select is absolutely essential for your Paper 5 and practical analysis. I know, nobody likes math and biology, but it's straightforward once you break it down. Comparing the means of two different groups, that's a t-test. Categorical data and comparing observed versus expected frequencies, chi-squared. Investigating the strength of relationships between two variables, correlation tests. Memorize this matrix. It's practically guaranteed points.
Mastering chloroplast pigments and technical methodology perfectly captures the need to deconstruct, solve, and evaluate experimental design. This is especially true when interpreting complex absorption spectra. A huge examiner red flag here is the confusion of cause and effect. Students often mistakenly say that the rate of photosynthesis affects absorption, when it's literally the reverse, and they use vague terms like "it absorbs light," instead of defining exactly which pigment, like chlorophyll B, is doing the absorbing at a specific wavelength.
All right, Section 5, Top Examiner Red Flags. We've talked a lot about what to do, but now we really need to look at the exact traps that are costing students their A-star grades.
These are the top examiner red flags. Avoiding them could literally save your grade. Simply missing units in calculations, like writing 3 instead of 3 micrometers per second, will instantly strip away your hard-earned method marks. Ignoring the command word is another classic mistake. If the question says explain, you cannot just describe what is happening, you absolutely must provide the biological reasons for why it is happening. Also, watch out for generic context, terminology confusion, and mixing up cause and effect.
Finally, Section 6, Final Exam Takeaways. Let's wrap up this explainer by consolidating everything into a final, actionable strategy you can use right away.
Remember this systematic sequence. Identify elements, apply principles, execute analysis, verify results, and present with precision. Always deconstruct the data first. Show all of your working to capture those method marks. Check your results for biological reasonableness. And finally, present your answer with exact, comparative wording and specific units. It's a mechanical process, really, and once you master it, the exams become incredibly predictable. I'll leave you with this challenge today. Take this exact 4-step framework, sit down with the past paper, and see how quickly your answers transform into top-band mastery. Ask yourself, are you still studying just to memorize biology, or are you finally ready to study the examiner? Don't just study hard, study smart. Thank you so much for joining me for this explainer, and I will see you next time as we continue to decode the secrets to your exam success.